Hello, my name's Christopher Bravery. I'm a consulting regulatory scientist at Advanced Biologicals. And today I've chosen the topic of control of critical reagents. So I've added the word analytical to make sure you don't get confused by raw materials, which are sometimes also called reagents. So critical analytical reagents. Why did I choose this topic? Well, partly because a client of mine, a biotech client, has had a couple of issues with their critical analytical reagents in the last year or so. And in their defense, they only took over the dossier at market authorization. They didn't write the dossier, but actually there were some deficiencies in this respect in the dossier, and then some mistakes made by their contract manufacturer. And it made me realize that actually, if anything, we probably have more critical analytical reagents for some of the advanced therapy products, um, not just viral vectors, where we probably have a more typical biotech-y type amount of critical reagents. But um, if you've got cell-based genetically modified cells, then actually you've probably got even more critical reagents than other products. And so, because it didn't really seem that I could remember that anybody had specifically spoken about it, and it is so important, I thought it was probably a good opportunity to introduce a topic that you might not have thought about, or not adequately yet, perhaps. So I'm going to cover what they are. What am I talking about? Are reference materials part of that? How am I supposed to control them? What are the dossier requirements and expectations at approval? And so overall, why does this matter? Okay, so I think the point to remember is that that your analytical methods are just as important to your product as, say, your raw materials. And the performance of those analytical methods is also highly dependent on the quality of the reagents that you're using. And this is the point to this talk, really. So why have you not heard about this before? Which some of you might not have done, some of you may have done. I think it's because, without going on a trawl through all the quality or CMC guidelines, I don't particularly recall it being specifically mentioned anywhere. It might be in Q2, ICH Q2, possibly. There is a bit on reference materials in ICH Q6B, but it's really very limited. And I think it may fall into the same category of the regulator not really seeing it as something that that they needed to write guidance on. But it is perhaps getting more attention, possibly because analytical methods are getting more complex. So where you tend to see any advice at all tends to be in guidances that you might not look at, because they tend to relate to clinical and non-clinical. In particular, anti-drug antibody, so immunogenicity assays. This comes up quite a lot because those are quite difficult to control sometimes. And that's sort of, and anything that mentions ligand binding assays, which also tends to be more directed towards non-clinical and clinical stuff. So it maybe is a little bit of a gap from the regulators, but hopefully I'll fill you in. So I've chosen to use a little bit of text from a new ICH document M10, which is bioanalytical method validation. EMA and FDA have guidances with pretty much the same name, which actually look remarkably similar. And in fact, PMDA have one that's really quite similar as well. But with differences in the text and varying amounts of text on this topic. So ligand binding assays are a good example of assays that will have critical analytical reagents. And they are basically anything where you use some sort of ligand to bind an analyte in order to make a measurement. So obvious things here are the problems that my client had with their host cell protein assay, for example, which was a process-specific one, but also things like potency assays. And of course, if you've got cells, if your product is a genetically modified cell, then all those flow cytometry reagents are all going to be in here. So let's look at some. Oh, sorry, going in the wrong direction. So the examples are things like flow cytometry, labeled antibodies, host cell proteins. So many of you might be, for your vectors, probably using an off-the-shelf kit from Cygnus, perhaps. You seem to be the ones that make them all. Those are a sort of generic host cell protein ELISA. And for some products, that might be acceptable all the way through to approval. But for other products, and generally this is done for biotech, they would make a specific assay that's specific to the process. And so you would take some host cell protein, some impurities from your process, and immunize rabbits or goats and create polyclonal antisera, which you then use to make an ELISA. You might label some of those with horseradish peroxidase or something similar, or a chemiluminescent substance or something, and create an ELISA. Now, just because you bought that ELISA off the shelf from a supplier, it doesn't preclude or doesn't exclude you from the requirements of dealing with these as critical reagents. It's just that you're now going to have to work with your supplier to deal with that. But other ELISA type methodologies, you might be using that to measure process-related impurities, or you might be using ELISAs to make some measurements of cytokines from cells in process or as part of a potency assay. Reference materials are described separately in the few guidances that mention critical analytical reagents. So I think they're not necessarily called critical analytical reagents, but actually everything I'm going to say or everything that you'll read when you look at some of the references later apply really to reference materials as well, plus a little bit more. And it's really not very well described in ICHQ 6b, and it's I toyed with giving a talk on that topic actually, but chose this one instead. So M10 is in draft at the moment. I think it might even still be available for comments. It's at step 2 of the ICH process, so it's not really come into force yet. It describes ligand binding assays as anything that specifically binds to an analyte. So you can see why things like ELISA are obvious examples, flow cytometry as well, but also enzymes. It could include things like restriction enzymes or other enzymes used for peptide mapping, if you happen to do that sort of thing, which most of you won't. But also the labeling of antibodies and so on as well would be included. So critical reagents are those sort of substances. Of course, it's up to you to determine if they're actually critical. But these are complex reagents where there is likely to be a difference batch to batch. So a typical IgG molecule does have a lot of variance, however you make it. A lot of that's down to the glycosylation, which is probably irrelevant, but there's other aspects of other post-translational modifications. So it can affect them, and it can affect their binding, and that can affect the quality of the signal you get. And labeling is pretty important too. It'll depend exactly what you're doing with the antibody and the label. So if you're looking at a low-level antigen, say by flow cytometry, then distinguishing a positive from a negative can be quite tricky unless you've got a good signal to give you a clean positive negative. Also looking at rare events, if you're making if you make CAR Ts and you want to check that there's no B-cell lymphoma left in the CAR T, for example, then you might be looking at fairly rare events. Or if you're looking for an IPS, but actually we're talking about gene therapy, so let's not drift too much into that, but they're easy ones to think of. So M10 says, reliable procurement of critical reagents is important, whether they're manufactured in-house or bespoke for you, you might have contracted that out, of course, or whether they're commercially supplied. So just because you buy a test kit, an ELISA to measure residual bovine serum albumin or a off-the-shelf host cell protein ELISA, that doesn't relinquish these responsibilities to evaluate these as critical reagents and ensure that you know how to manage them over the product life cycle. Now you'll note I've highlighted early in development, and I think that's an important point, that you really want to start thinking early. And I know that everything should be thought about early. If you've got a compressed timeline, then you definitely need to get on with it ASAP, because you really need to have all the processes in place, and you're putting probably quality on a critical path by doing a fast to say conditional approval type approach. So some of this will come out of some early robustness studies, but then you need to think about the management over the life cycle. How are you going to swap a new batch of reagent when it comes along? How are you going to reevaluate it? Does that require partial or full revalidation of the analytical method? What should be in the dossier? Well, the dossier should identify this as a critical analytical reagent, and it should provide adequate information to identify that reagent. So that might be the name of the reagent, might be a catalog number, perhaps. Batch lot is all a little bit like a critical raw material. Things like stability important, and making sure that you record these sort of batches in key reports, etc. Again, this table that I've taken from M10 is sort of assuming that this is a sort of a clinical assay. And of course, if you do clinical assays, then, this is also applicable. So in fact, I'm adapting it from that, because there's very little written for quality assays. And importantly, you really need to establish up front, ideally within the market authorization dossier, how you go about these changes. These aren't changes that you can just do and get on with it. They are reportable changes. In many cases, they might be a type 2 in Europe or a 60-day procedure in the US, or you might get away with a 30-day procedure. I didn't sort of go and have a look and think about which ones would be which for this, because there wasn't going to be time in this talk. Criticality, well, like anything else, you can evaluate criticality for yourself around probability of, things going awry and impact. And I think you'll agree that an antibody that doesn't quite do what it doesn't quite bind as well as another antibody, or has a poor labeling or something, it is going to affect the result of your assay. It might reduce the sensitivity in the range, for example. So it is up to you to identify that. Some of the publications I identify sort of suggest which things may or may not be critical, whether they're always critical or sometimes critical. But many of those examples aren't entirely useful for this talk. Stability, you need an ongoing stability program. You need to keep an eye on it. This particular paper from the AAPS journal even provides some sort of suggested periods. But obviously take this with a slight pinch of salt and make sure that you evaluate yourself. which means you're going to have to come up with ways of evaluating it and looking at it. This is not particularly trivial, unfortunately. So what you're trying to do is obviously maintain consistency of reagents over time. And you might be on the market for 20, 30 years, maybe more if you're lucky. And if the assay doesn't need to change, or you don't change the assay, then you're going to have an ongoing requirement to keep that assay performance up to scratch, which might mean things like trending and things are useful of your analytical methods in the same way you do with your process. And it will be expected that you compare the existing batch of critical reagent with the new batch of critical reagent. And there's a little bit in there about what might be a major or a minor change. I think it should be fairly obvious. It depends on your evaluation of criticality, I suppose. So there's this is really just mostly to identify a paper that you might find useful to read. It gives a nice little sort of life cycle diagram that I thought looked quite pretty, but I've realized I don't really have time to talk through. But anyway, you get the point that in the same way as you manage the life cycle of your raw materials, you've got a life cycle of your critical reagents. What I do want to pick out is a problem I have seen several times, and not just necessarily in a critical reagent aspect, but in other aspects like reference materials. So, and indeed, this was the problem that my client had with their potency assay critical reagent as well, that the supplier, the contract manufacturer had started off with the first batch. And then when they'd had to get a new batch, they'd compared it to the first batch. Well, that bit's fine, of course. But when it came to the next batch, they compared it to the second batch. And then when it came to the fourth batch, they compared that to the third batch. And as this figure tries to present, there's a great potential, if you do that, to get serious assay drift. So I really don't recommend that approach at all. Better to use the approach that you would do with a reference material, of having a reference critical reagent batch, if you like, keeping a batch from some material from the first batch, assuming that it's got adequate stability that you can do this. Of course, it may not be able to do it over a very long period of time, but the longer you can do it, the more more stable, hopefully, your results will be. So there you compare the second batch to the first batch, but also the third batch to the first batch, and then the fourth batch to the first batch. So hopefully you're not seeing drift so much as just wobble. I hope, anyway. So that would be the idea, to try and minimize that problem. So as I say, there isn't a huge amount of literature out there. So there's EMA, FDA, and actually PMDA, all within a reasonably not very long in regulatory terms period of time, have written guidelines on bioanalytical method development, which I bear quite a lot of resemblance to M10. You can see that this is consolidating guidances written by different agencies that are actually quite similar. The EMA just have a section on reagents, says very little really, but at least hints that you need to deal with your critical reagents. FDA one, bit newer, so it actually uses the term critical reagent and provides a little bit of text, and it distinguishes reference materials from critical reagents, although why you wouldn't just call them all the same thing, I'm not sure, because it's still a reagent of sorts. USB, well, as I say, this issue, I think, has been primarily brought up by regulators in guidance related to anti-drug antibodies. So USB 1006 says a little bit under life cycle management, probably not enough to go and have a look. So I've listed a couple of papers at the bottom. The EBF recommendation, blah, 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 is for anti-drug antibodies as well. But if you read past the anti-drug antibodies, you'll find some of the advice in there quite useful, but you just have to modify what they mean by it, of course. And then there's in the AAPS journal, there's quite a nice article on ligand binding assays, which talk about critical reagents as well, from 2012. And thank you for listening. And I hope you found that useful. And thank you.